from Alt-Jan, I believe, on integrating machine learning and mechanistic insights. So, thank you, Alt-Jan.
Yeah, okay, thanks. Happy to be here, so thanks for the invitation, in particular to the students and Yingti in particular. So I'm going to talk about some ongoing work in my group, in particular 2 examples where we try to connect machine learning approaches and mechanistic models. And the context here is that these are projects in the CropXR program, which is aiming at breeding for resilient crops. Later today, this afternoon, Kirsten ten Tuscher will talk more broadly about this project and give you an overview. So in my talk right now, I will just present these 2 examples that we're working on in my group. And one aspect that is good to know about this CropXR program already is that it's a collaboration between breeding companies and academic researchers, and then both experimental researchers and computational research. And one of the specific aims for the computational people in the project is to work on methods for connecting indeed mechanistic models with machine learning and deep learning based approaches. So I have this slide that I think I can go over very quickly because I'm sure most of you are aware of this, but just to remind you, we're talking about, on one hand, machine learning, which is a subset of AI approaches, so approaches that are data-driven, and where the focus is to find patterns in your data and use these patterns to make predictions. That's on the one hand. And then on the other hand, mechanistic models where you would use knowledge that you have about your system to construct equations that describe certain processes in the system. And this I'm showing here is an example of a model that I worked with in the past. Each of the 3-letter or 2-letter abbreviations are genes. And this model then explains how certain genes influence each other. And in the examples that I show today, we'll also focus on, indeed, models that work at the level of genes and particularly gene expression. But of course, very interesting to think about expanding that. So why would we want to combine these models? Well, it was mentioned already that actually the question that was just asked is a very good drive towards this idea of indeed combining these 2 modeling approaches, because each of them have their own strengths and limitations. For machine learning models in general, you need a lot of data. And with mechanistic models, you would be able to maybe compensate for that by using knowledge that has been obtained maybe already over decades. So there's a lot of interest, not only in biology in general, but also definitely outside, to indeed try and combine mechanistic models with machine learning models. And I think it's very appropriate for us in this field to also make use of those developments. So what I will do now is to indeed present 2 directions that we're working on, and this is really work in progress, so I'm not showing a lot of definitive result, but I hope it will help to get you thinking on the possibilities in this area. The first example is on an approach where we have this idea of first using unsupervised machine learning to make groups of genes, so clustering, and then using a mechanistic approach to make connections between the clusters. And this is based on the fact that clustering gene expression data is something that is done a lot. It helps you to find groups of genes that are relevant for certain processes and that have related behavior, but these clusters are typically static. There's no dynamics involved in there. So in this approach, what we're doing is we now are at the stage where we have indeed a proof of concept. We start with a time series of gene expression, and we find gene modules by using, in principle, your favorite clustering approach. So this is also part of the research to try and see what clustering approach works best. We can connect also with existing data sets where co-expression is used to find clusters. So it's a way to integrate newly measured time series with existing gene expression. And then we have a couple of steps that, in view of time, I don't think I can discuss them now in detail, but these allow us to make a selection of most relevant gene modules and also predict connections between the different modules based on the presence or absence of certain transcription factor binding site in promoters of genes. And then we have a final step where we fit a differential equation model to describe the average dynamics of the genes in each module using these time series that I mentioned at the beginning. So I'm showing here at the bottom two very preliminary results of this approach, which are based on measurements in Arabidopsis, either with draw treatment or with heat. And then in both cases, at the left-hand side, you see a representation of the obtained modules. So each yellow circle is a set of genes that are clustered together. The arrows are the predicted interaction between those modules. And then the time courses here indicate the fit of the ODA system to the average dynamics of the genes in those modules. Okay, that is the first example, and I will now move on to the second example, which is on using again a combination of machine learning, in this case deep learning based classification, with again ODE models. In this case, the idea is that we want to use deep learning to predict gene expression based on promoter sequences, and then use the predictions of those deep learning models as input for an ODE model, in this case for the description of enzyme dynamics. So I'm starting here with an overview of the deep learning model. In the plot here in the middle, you see example of data that we use to train the model. Here we have measurements in different conditions, different treatments with different hormones. So that's each of the 6 panels over there. And then the blue and the red indicates expression time courses for 2 genes. Of course, we have these for all the Arabidopsis genes, but these are just examples of the expression dynamics of 2 genes in these different conditions. And for now, we convert this to a classification problem by first detecting differential expression for each gene between each condition and the control condition. So that results in a table like this, where for each gene you get, for each of the different treatments, a 0 or a 1, indicating whether the gene is differentially expressed or not. This is for now to keep things simple. We can also think of using a more regression-based approach, for example, but for now, this is what we use. And then the deep learning model is trained to predict these differential expression status for each gene based on the promoter of the gene. This is an example of the prediction results. For now, we're at the left-hand side, you see the ROC curve for these different 5 hormone treatments. So each color indicates one condition that we make a prediction for. And at the right-hand side, it's the same 5 models, but then it's a calibration curve. And for us, this is a reasonable result at the moment. So we know from literature that this kind of prediction can be done with reasonable accuracy, and indeed this is what we see here as well. And then the next step, and this is something that we still need to do, but the next step is that we are developing an ODE model for the dynamics of certain enzymes involved in a pathway that I'm afraid is not really well visible for you, but this is about the production of glucosinolates. So what the measurements involve is concentrations of different compounds in the pathway, and what this mechanistic model in principle can do is to predict those levels, for example, in mutants. So here we see a comparison of measurements and model predictions. And what is now the idea, and this is not something that we've done yet, but what we will do is to connect the deep learning-based prediction of expression levels as input for the ODE model, because this is a model where explicitly enzyme concentrations are present. So at the moment where we can predict indeed gene expression levels with our deep learning model and how these change upon different treatments or upon changing the genetic background, then we can also use that as input for our ODE model. So this is what I just said, that indeed we will be able to connect these 2 models, and at the moment we can do this because the genes that are present in our deep learning model, these are the same proteins that are also present in the ODE model. So this is a case where data or model integration is very straightforward because we have the same entities in the two models. It's of course also very well possible from data fusion type of point of view to think of combining different models that don't deal with explicitly the same set of components, and this is something that we're thinking about to work on in the future. So maybe just to clarify, I also don't know how much time I still have. A few minutes, okay. Then I will go a little bit fast, but I just wanted to clarify, how do we actually take into account environmental or management inputs in this type of models? And this is indeed different in the different types of models. In the ODE models, in the first example, it's just a specific input term in one of the equations in the ODE system. Whereas in the deep learning example, the second example where we predict gene expression based on promoter sequences, the environmental or management inputs is currently presented as by representing multiple different classes for which we predict the expression level. So we have treatment A, treatment B, treatment C, and we predict expression for each of those treatments. And at the moment, these treatments are independent from each other. There's no knowledge in the model how these treatments relate to each other, but it's very well possible to think about ways to also have an explicit representation of these different treatments so that there will be more explicit ways in which the model can learn how one treatment relates to the other treatment, let's say. And of course, as I mentioned, this so far is only work in Arabidopsis, but within the larger context of the Crop XR project, for sure, we'll also start working on relevant crops. And in that direction, for now, we started looking at data in tomato, where this deep learning-based expression model that we trained with Arabidopsis data, we now also tested it on promoter sequences in tomato. And I was kind of surprised to see that even without fine-tuning the model, so just by applying the model trained in Arabidopsis on tomato data, we already get some performance, and this is quite promising, I think, because we will be able to further fine-tune this model and improve it also in tomato in this case. So to conclude, I talked about how we work on integrating machine learning and mechanistic models, and currently this focuses on Arabidopsis and the dynamics of gene expression. But I see a lot of opportunities to further expand this. I also want to make a little bit of advertisement for a review that we recently published on this idea of combining mechanistic models with machine learning, where we use the term scientific machine learning, which is sometimes used to describe this idea. And then I want to thank the people involved, so some local people here from Wageningen, Dick and Ben, people in my group at University of Amsterdam. You can see some of them here, and also collaborators from Utrecht University who actually provided the data that we used. And thanks for your attention.
All right, thank you, Alt-Jan. I think again we have time for one quick question.
Hi, Christine Beveridge, thanks very much for that. I was just wondering, with your ODEs, how many genes can you represent in those networks?
Yes, so currently that is not a lot, so maybe I don't know, 20 or so. That's also one of the reasons why in this first approach we focus on this idea of making clusters and then representing the dynamics of clusters, because that reduces the problem a lot, of course. And also in the second case, this pathway maybe contains 20, 30 genes max, I think. So for the moment, we are considering relatively limited ODE systems. But yeah, there is people in the field that think of much bigger systems. So might be interesting, but not something we currently do. No.
All right, thank you, Alt-Jan. And then we'll move on to our next speaker, Istvan.